My name is Manish Gupta, and in this video, I'm going to talk about AlphaFold. How can you use AlphaFold to generate highly accurate protein structure prediction, right? This is super important because AlphaFold has become very, very popular in the computational biology community. I think it is probably one of the most popular deep learning models in computational biology, right? And it has solved a problem that has been existing for a few decades, right? Therefore it's a very important thing. So what is AlphaFold and why is it important? Let me sort of talk about the original problem, and then we'll see how AlphaFold has solved that problem. Okay. Now all of us know that the way we behave, the way we look, the way we are is basically defined by the genes in our DNAs, and those genes are basically created from proteins. Now, these proteins essentially, there are so many proteins that are available, and their function really depends on their 3D structure. So proteins basically contain amino acids, and they are linked to each other, and how they link with each other defines their shape, and their shape really defines their structure. Their shape really defines their function. What do they do, right? So for example, here are some examples of proteins and their functions along with their structure. So antibody proteins are basically Y-shaped. They are more like hooks, and that helps them latch on to virus and bacteria, and that is what helps them detect and tag those disease-causing bad guys, the microorganisms, and delete them or exterminate them. Right? They're collagen proteins. They're shaped like cords, and they help transmit tension between various components like cartilage, ligaments, bones, and skin. Okay. Cas9, they act like sensors. They act like scissors to cut and paste sections of DNA. Right. Antifreeze proteins, whose 3D structure sort of allows them to bind to ice crystals. So basically saying, and prevent organisms from freezing and so on. Right. So basically saying different proteins have different structure, and therefore they basically have their own function. Right. So basically, and the nice part is that we have been obtaining amino acid sequences for a lot of proteins thanks to efficient genome sequencing. In the past decade, genome sequencing has become super efficient and super cheap, right? So therefore, we have like lots and lots of proteins which have been sequenced. Basically, sequencing means we know their amino acid components, but we do not still know their structure. Okay, that is the problem. So the problem is that finding the 3D structure from the genetic sequence, from the amino acid sequence for a protein is complex. Okay. Now, so basically you have proteins, the amino acid sequences, but you don't know their shapes, right? And if you don't know their 3D shapes, you don't know what they're good for, right? It's like basically having tools, but I don't know how to use it. That's the problem. Or basically I have a problem and I don't know which of these, millions and billions of proteins that I have will solve that problem, right? So knowing the structure is super important. It's super important not just for finding, more about ourselves, more about, how do they behave inside human body, but also for designing proteins which can solve some problems which are popularly known, but we don't have solutions for them. So for example, knowing structure is important for diagnosing and treating some diseases which are believed to be caused because of misfolded proteins, misfolded in the sense that their shape has gone bad, right? So if you can correct their shape, maybe you can solve these problems like Alzheimer's, Parkinson's, Huntington's, and cystic fibrosis, right? These are diseases which can be solved, which are believed that they can be solved if you can repair the protein structure, right? They can, now proteins could also be used for other purpose, like figuring out how can you synthetically generate biodegradable enzymes that can help make the waste material biodegradable, like plastic and oil biodegradable. Okay. Finding structure, however, as I said, is very, very complex. So the current biological methods that are available and chemistry methods that are available, they basically take years and can cost tons of thousands of dollars to find out one structure given a protein. Okay? And that is why people are looking into machine learning, deep learning ways of synthesizing the 3D structure, figuring out the 3D structure of proteins. Okay. Of course, figuring out the 3D structure is super important for drug discovery, right? So figuring out what important, protein can be used for what kind of solving what kind of disease or countering which kind of bacteria and viruses. Okay. AlphaFold is the first deep learning method that can regularly predict the protein structures with atomic accuracy. Atomic accuracy meaning, it can actually predict the positions of these atoms nicely. And it's super important because actually it won the 14th CASP challenge, which is basically critical assessment of protein structure prediction. It won the challenge by a significant margin, and that is why it has become super popular, right? So this is how basically, the protein structures look like. These are three different protein structures. And as you can see, the blue one is essentially the predicted structure, and the green one is the actual structure, ground truth structure. And you can see green and blue are very close to each other, meaning AlphaFold can actually do a pretty good job at figuring out those proteins. Let's talk about how accurate is AlphaFold, objectively. So basically, in this world, basically, you're trying to predict the protein structure. So essentially, you're trying to predict the positions of various atoms. Remember, there are these carbon, nitrogen, and various kinds of atoms inside a protein. So you're trying to predict their structures and the atom positions of those atoms. Now, you try to measure those positions of the atoms and then predicted positions versus accurate versus the actual true predictions. You try to figure out the distances between them and define a metric. That metric, if you really look at only the alpha carbon atoms, right? So there are some atoms which are called alpha carbon atoms, the main atoms in some ways in a polypeptide chain. So if you try to compute their root mean square deviation, this is what is actually plotted in this first graph, right? And as you can observe, the G427 team, basically, which is the AlphaFold team, essentially gives you the lowest root mean square deviation. The lower the better, this metric. And that, compared to various other teams which participated in the CASP14 challenge, right? So you see, amino acids are the building blocks of these proteins. And in these amino acids, the main building block, the backbone, is actually called as N-C-alpha C backbone. N is nitrogen, C-alpha is the alpha carbon atom, and the C is the carbon atom attached to the alpha carbon atom. Okay? And what is each amino acid? Well, each amino acid basically contains multiple chemical groups, which we studied long back in chemistry. Some of you might still remember: amine group, NH2, carboxyl group, COOH, and hydrogen atom, and a unique side chain, which is also called as the R group. But we'll call it side chain in this video, right? Which is attached to the central carbon atom. So now you see, of course, these proteins are basically sequences of amino acids, right? So essentially, when you have an amino acid, you basically, it undergoes a chemical reaction to become a part of the protein chain. And when it becomes a part of the protein chain, it's called as a residue. Okay. So essentially, it's called residue because, in this chemical reaction of becoming a part of the protein chain, it loses a water molecule, and then what remains is called residue. And that is why basically, amino acids and residues in the protein are the same thing. It's just that when they become a part of the protein chain, they basically are called residues. Okay. So basically, proteins basically are polypeptide chains. Basically, they contain multiple. They basically contain multiple amino acids, and each amino acid basically has these groups where the main backbone is formed of N-C-alpha C, and then there are side chains, as they're called. Basically, all of these other groups, they form side chains, and these side chains, of course, can influence. In fact, they can influence the dihedral angles, as they're called. So now there's an N, C alpha, and C. Now these three atoms, the angle between them are actually popularly called as phi and psi. Okay, they are given those specific names, and they're called as dihedral angles. So, and these side chains can actually influence these angles between N, C alpha and C alpha C. So the angle between N C alpha is called phi, the angle between C alpha C is called psi, and then depending on the side chains which are associated based on the amino acid, each amino acid is a different unique side chain. So depending on these side chains, the angle phi and psi can differ for each of those backbones, in the protein chain. Okay. Now, when you think about these side chains, they themselves will basically have their own dihedral angles, and they are called as chi angles. So, chi1, chi2, chi3, chi4, some of them also have chi5, but most of those amino acids actually have chi1 to chi4 dihedral angles. So basically saying whenever you have a sequence of three atoms, there are going to be bound to be two angles, and those two angles basically, are called as the dihedral angles. The dihedral angles of the side chain are called as chi1, chi2, chi3, chi4. Okay. The width of a, so, if you look at it, the root mean square deviation, right, you would observe that the root mean square deviation obtained by AlphaFold is basically less than 1 angstrom. It's measured in the units called angstrom. Now, to compare, the width of a carbon atom is basically 1.4 angstrom. So basically saying that AlphaFold can actually predict the position of the carbon atoms for new proteins of the C alpha, carbon alpha carbon atoms for these new proteins with atomic accuracy. Basically saying the root mean square deviation is less than the width of a single carbon atom, okay? Which is beautiful, which is really very important. Okay. So basically what you can see here are these two proteins which are shown in this figure and the figure on the right side. So basically the predicted one is the blue one and the true one is the green one. So, and as you can see, it basically can predict with very good accuracy, not just for smaller chains, but this particular one on the right side is basically contains 2180 amino acids or 2180 residues. And look at it, I mean, the accuracy, basically root mean square deviation is 2.2 angstrom, which is pretty good in that sense. Okay. So further, how accurate and reliable is AlphaFold? So, you can see this is basically the distribution of fraction of proteins with respect to the root mean square deviation. Right? So you can see the C alpha root mean square deviation for a whole bunch of those C alpha atoms. The root mean square deviation is less than 1.4, which is basically this one, right? Which is basically less than the carbon atom's width, essentially, right? So for a few, very few of those atoms, yes, I mean, sometimes it can make larger errors, but mostly it basically gives you smaller errors for most of those carbon C alpha atoms. Okay. Now, you could basically also compute accuracy in terms of the backbone prediction, how accurately you could predict the backbone versus how accurately could you predict the position of the atoms in the side chains. Okay, so on the x-axis, what you see is the accuracy of the prediction of the backbone as measured by the score. We'll look at the definitions later, IDDT, but some metric, right? This metric, the higher the better. So essentially, you can see that IDDT C alpha. Basically, oh no, sorry. So this metric is also a distance metric. Sorry, the lower the better. But what you can see is that x-axis basically is the backbone prediction accuracy. Y-axis is basically the side chain prediction accuracy. You see there is this chi1 angle which is being measured here. So the accuracy of the side chain, they are basically measuring in terms of the chi1 angles. So basically, the x-axis backbone accuracy, y-axis side chain accuracy, and what you see is that there is a very good correlation. So if basically saying if you basically accurately measure backbone prediction, then the side chain prediction is also accurate. Okay, which is good. I mean, in that sense, it's for a system. The third graph here basically shows you, that the predicted, on the x-axis, basically the predicted metric, so predicted local distance difference test metric. On the y-axis, basically what you see is the actual metric. So if you basically take the actual protein structure and you compute these distances between these various atoms, C alpha atoms in the entire backbone of these structures, y-axis actually is the metric which is original on the ground truth molecule, right? X-axis is on the predicted molecule or the predicted distances, and what you see is that there is a reliable prediction mainly because there is a linear correlation between the two, right? Even in this dense region, you basically observe that there's a linear correlation, basically saying that not just that the system AlphaFold can actually come up with very accurate predictions, but it also knows when it is not accurate. So basically, this metric actually tells about the confidence of the prediction. So the confidence of the prediction is also very accurate in terms of whether it is coming up with a good protein structure or not. So if the confidence is low coming out from AlphaFold, you can actually reject those protein structures as identified by AlphaFold. Okay. Now let's talk about how does AlphaFold work. So, in this case, this is the overall architecture of the AlphaFold system. So there's of course a lot of detail in this system. It's actually very complicated. So when I started reading this, I sort of got so involved. It's a very, very complicated system. So let's basically start from the input sequence. Essentially give the amino acid sequence, and that's all you have. Now these amino acid sequences can be very long, and therefore at train time, you essentially divide them into fixed size sequences and just give that part of the molecule, that part of the protein as input. Okay. So one sequence is not enough. You want to basically depend on the structure of similar sequences, which you already have figured out structure for. So therefore, you look up the sequence in a genetic database search. So basically, you look up this input sequence to get nearest neighbors from two different databases. One of the database is genetic database search, and the other is the structured database search. Okay, so as the name says, you're basically going to find nearest neighbors, other sequences. So as you can see here, these are other sequences which are similar to the sequence. And you're also going to find other sequences from the structured database search. But there's a difference. Let me talk about the difference. In the genetic database search, you're basically going to find out those sequences which have similar amino acids. So if there are some kinds of amino acids, and by the way, basic biology. So if you didn't know, proteins typically have 20 amino acids, 20 popular amino acids, and then others are unknown in that senses. Okay, so that's some sort of like 21 possible units in this input sequence. Okay. So you do a match with the existing protein sequences, and there are crazy number of protein sequences which have been sequenced anyway. So you basically get those matches and find the ones that match the most. Okay? And there are nice efficient algorithms to do this. They're called as local alignment algorithms. One of them is Smith-Waterson algorithm and so on. But those are in computational biology very popular. You can think of them as a black box. And there are methods like FASTA and so on, which are basically used to figure out these matching sequences. Okay? So basically, these are basically called as MSAs or multiple sequence alignment. So for every input sequence, you get aligned sequences in that senses. Okay. Now you also basically take the sequence and search it on a structured database. So structured database basically the database where you have the sequence and you also have its structure known. The the atom structure, the 3D structure is already known. Okay, so you basically take this sequence and you do a structured database search. You basically figure out those sequences which match a lot, and you basically take their 3D structure and you essentially get this pair amino acid or peptide pair kind of interaction matrix. Okay, so here you basically get matching sequences, but here you do not really get the amino acid sequences. What you want to get is these pairwise interactions. Okay, so you get the pairwise interactions between each of those residues. You'll start calling them as residues. So a protein is a sequence of residues. Okay. So essentially, if you look at this MSA matrix, basically it has a number of sequences as the rows and number of residues as the column. So it's a sequence cross residues matrix. Okay, we'll call it as S cross R matrix. And then, of course, for every amino acid, it basically has some features. So basically, therefore, it's a tensor S cross R cross tensor. But what you also have is this tensor, which is basically an R cross R tensor, because if you have like R residues, it's basically showing you the interaction between R residues with each other. Okay, so what it does is to basically take those residues, and then it basically is trying to figure out paired features. So residue paired features. We'll basically talk about what are those features into more detail later, but it basically looks at those pairs of residues, and it figures out those paired features. Okay. So in short, basically, if you look at the remaining part of the architecture of AlphaFold, you basically give it like 3 different inputs. One is the input sequence, the other is these MSAs or multiple sequence alignments, and the third are these pair representations, also called as templates. Okay. That's that. And what does AlphaFold do? Well, AlphaFold at the end of it, and of course, it comprises of these blocks. We'll talk about them. By the end of it, it's basically going to give you the 3D coordinates of all the heavy atoms for a given protein. Heavy atoms basically means atoms like nitrogen, carbon, oxygen, and things of that kind, right? It's basically going to give you these coordinates of those heavy atoms for a given protein by using the input sequence, the main amino acid sequence, and aligned sequences of aligned similar sequences, basically. Right? So AlphaFold basically comprises now of 2 important blocks, evoformer and structure module, right? Large blocks. Of course, they're transformer-based blocks, 48 blocks here and 8 different transformer layers here. And we'll talk about how there's a lot of novelty in these layers, by the way, a lot of complexity. Okay, we'll talk in detail about those, but it basically comprises of these 2 different stages in some ways, evoformer and structure module. And then interestingly, there is an iterative refinement which keeps happening here. And this is basically what they call as recycling. So they recycle 3 times. So it's basically pretty deep architecture, 48 blocks into 3, basically, which is recycled. So that's that. And then there are 8 other structure module blocks, basically. Okay. So that's that. Now, AlphaFold actually does 3 things mainly. So of course, it figures out the 3D structure, but it does it in 3 parts. First, it figures out the backbone. So it tries to figure out, remember, a protein basically consists of several residues. For each residue, it figures out one backbone. So one or more backbones, sometimes it could have more backbones, but primarily one backbone. So for each protein, it's trying to figure out those backbones in C alpha C. So those are the backbones. Okay. Second, it basically tries to, and by the way, figuring out backbones basically means figuring out their positions in atom, C alpha atom and C atom, and also figuring out those dihedral angles, okay, the phi and psi. Okay. Then it tries to figure out side chain placement. So it basically tries to figure out where in this amino acid sequence I should place the side chain and the positions of the atoms in the side chain and the dihedral angles there, so the chi angles, right? And then the third part is, of course, it through this iterative framework, it keeps refining through backpropagation. It sort of keeps refining the positions to ensure the predicted structure is sort of physically possible. Remember, you're predicting structures of things, positions of physical things. So, for example, if you're predicting that two atoms are at the same place, of course, that's not a plausible physical situation, right? So, therefore, it basically tries to figure out those physical constraint violations and then tries to refine the structure until it gets a really good structure. Okay. So now let's think about how do you give these inputs. Remember, I told you that you have an input sequence and you give like two kinds of inputs to it. One basically based on this genetic database search, the other based on structure database search. How do you figure out these inputs, right? Let's go into a little more details. Okay. So basically the inputs, as I mentioned, to AlphaFold structure, the AlphaFold architecture are these: the primary sequence, which is the input sequence, the sequences from evolutionary related proteins, basically which have a very common amino acid sequence, right? And they are basically these multiple sequence alignments. Okay. So remember, when you do multiple sequence alignments, you would not find exactly matching ones, right? You'd find slightly matching ones. So when you do this match, you basically use an edit distance kind of an algorithm. It's like saying, yeah, I have a sequence and you have another sequence and you want to do edit distance, but you want to figure out those sequences which have lower edit distance, but you also want to do not global alignment, but local alignment. So as we evolve, right, we have copied the protein sequence, the genetic matter from various organisms, and that's how organisms evolve. Right? So when you do this local alignment, remember in edit distance kind of computation, you have to insert, you have to delete, and you have to substitute various items, right? Various units. So here also deletion happens. And so therefore, some gene is deleted or some amino acid is deleted, some amino acid is added and so on. Okay. So in these matches, basically, you would therefore have gaps sometimes, basically, which represent deletions. And, sometimes you would have like inserted things and so on. Okay. So basically, you have these MSS multiple sequence alignments, and you also have these paired representations here called templates. Okay. Those are the inputs. And you pre-train on 256 residue kind of sequences. So essentially, they do two things for their model: pre-training and fine-tuning. And at pre-train time, they essentially, they use a bunch of losses which are defined without any labeled data. But then fine-tune time, you have to basically define those losses based on 3D structure labeled data. Okay. So at pre-train time, the sequence length is 256 residues. That's basically like the number of tokens in our world, right? In a typical NLP world. Okay. And they pre-train with 10 million, more than 10 million samples, that because this data is available in plenty. Okay. They take like 1 week on 128 TPU V3 cores to basically train this model, pre-train this model. Fine-tuning basically is done with the longer sequences of 384 residues, and they take 4 days on 128 TPU cores, V3 cores to be able to fine-tune this. And we'll talk more in detail about each of those processes. Okay. So after they have trained the entire network, this is basically just first stage. So after they have trained the entire network, basically, which means all of this is done, they essentially run this network on another large data set called as uniqueness data set, which basically has 356K sequences. So what they do is to run this network on that data set, and they make structure predictions. Now, high confident predictions, they basically take them back and use them to retrain the entire network. Okay. So this basically, it used to be called as self-training in the previous world. They just call it self-distillation here. The idea is basically you take a whole bunch of unlabeled data. You essentially, get the data labeled using the same model, using the first version of the model, and then you essentially use it for training the second version of the model. And the second version, they basically take 75% samples from this self-trained data and 25% from the original PDB data, the protein data bank data. Okay. That's how basically they do self-distillation or two-stage training. The first stage basically comes up with some predictions, which is high confident ones are used in the second stage. Okay. And not just that, they do even more rigorous work. They train ensemble of 5 models. So remember, it takes like 1 week for 128 TPU cores. Well, 2 weeks because you're training it 2 times, and then multiplied by 5 because you really require an ensemble of 5 models. Okay, so lots of compute requirements. Now, you can come up with many sequences which are very similar to the input sequence, but then, you may not be able to use all of them because maybe in a batch you only want to fit a few of them, right? For efficiency reasons. Therefore, what they do is to cluster these MSAs. They basically say, hey, let me cluster a few of them and take diverse such sequences. Okay. In fact, even for more diversity, they also basically find these sequences which are very similar and delete some blocks so as to have a lot of diversity. Otherwise, what you would do is that if you've taken a human sequence, you would probably take other human sequences which are very similar, and therefore the 3D structure that you learn at the end is going to be very biased. And probably you don't want to learn so much of bias. You want to have diversity in them so that the training can be done better, right? And therefore, they basically do clustering and some sort of removal of the redundant ones when creating when getting this set of nearest neighbors in terms of other aligned sequences. So by the way, they cluster these things, and then when they cluster, they may find some sequences are similar to the input sequence, but they don't really belong to those clusters. And then they call them as extra sequences. So there are these clustered sequences, which are called as n-clust. So n-clust number of clustered sequences. And then there are these sequences which form templates, essentially, which help get you paired representations. But then there are also these other sequences which they actually call as extra sequences, okay, which are also part of the MSA structure, but they are basically not a part of those clusters in that senses. But yeah, if you look at the MSS representation, they have the cluster ones plus the extra sequences both. Okay, of course the templates form a part of these paired representations. Okay. Okay, so that's that. Now, specifically, if you really observe, the MSA matrix is basically a number of sequences. The order, the dimensions is number of sequences multiplied by number of residues multiplied by the depth, which is number of channels. Right? So what is the number of channels? Basically, you require some features for each of those amino acids here, right? Here, the pair matrix, if you see, it's basically RRC, which basically means it's a number of residues, cross number of residues, and depth is number of channels. So what are the features that you use in the pair representation? What are the features that you use in the MSA representation? See, MSA representation is SRC, number of sequences, number of residues, and number of channels. So what is it? What are the features here, right? So there are lots of features, and the right side picture actually shows you how those features are obtained. Okay, so as you can see, there are these various kinds of features, raw features that you get, and then there is all of this neural network manipulation which is going on, so as to be able to give these features finally to the evoformer stack. What was evoformer? Remember, that was the stage one. Evoformer was the stage one of our model. Okay, and then the structure module is the second stage of the model. But anyway, we are currently worried about how is this input provided, right? So as I mentioned, the input has to be basically featureized a lot before it goes to the evoformer part of the stack. Okay, so there's a lot of processing which is going on here. Okay, so as I mentioned, there are three things mainly. One is the original input sequence. Second, there are MSAs, which are aligned sequences, and the third is the pair representation. And they're going to be crazy number of features which capture the information from these now. Okay. So just a note, I mean, R is the residues, F is the features, C is the channels depth, right? Now, specifically, SC is for clustered MSS sequences. So SC is the number of clustered MSS sequences. Of course, S is the overall number of sequences, but SC is the clustered MSS sequences, SC is the extra MSS sequences, ST is the template sequences. Okay, that's basically that. So let's essentially talk about how are these inputs obtained and what do they contain. Okay, if you really look at just the original sequence, let's call it target feature, the original sequence, right? The single protein sequence. It basically has amino acids, it's a sequence of amino acids, right? So what can you obtain from that? So the original, and remember, I mean, at pre-train time, you have 256 residues. So it's a sequence of 256 different amino acids. Okay. So this sequence is really represented using these AA type features. Now, what are these? These are 256 multiplied by 21 features. That's a large number of features by itself, right? What do they contain? So they basically contain one hot representation of every input amino acid. Okay. Now, as I mentioned, there are 20 amino acids, and then there is an unknown thing. So some units are called as unknowns in these proteins, right? So practically every unit is basically one of these 21 different classes. So you encode them using one hot representation, and you encode each of them, thereby coming up with this feature called as amino acid type, which is like 256 into 21. Okay. Now, for every MSA sequence, which is basically clustered sequences or extra sequences. So let me talk about the clustered sequence first. So for every MSA clustered sequence, you actually have similar kind of features. Okay. So every MSA clustered sequence, so therefore you have number of clustered sequences and you have number of residues because of course all of them also have 256 residues. So that's that. And you have like a one-hot representation of every residue there. Now, this one is 23 and not 21 because beside these 21, there could be gaps and there could be something called as masked tokens. Okay. So at pre-trained time, remember, in BERT, you have those masking which is done, and then there is an MLM loss that people define. Actually, they reuse the same loss here. And therefore, sometimes they mask certain amino acids, which is going to be predicted at the end anyway. Okay. So, and that is how one of the pre-training losses actually. So therefore, in these clustered MSC sequences, there could be, of course, the 20 to 20 amino acids are unknown, and then there could be a gap, which is basically because when you're doing alignment, you basically deleted some gene from here, right? That's basically the gap. And then masked MSA tokens, okay, masking. That's basically 23 different, things in the 100 representation, and you have like residues, so many residues. For every residue, and then for every clustered sequence, okay? So that's that. Now there are many other features. Of course, given this, given that I don't want this video to become several hours, I'll not basically go into details of those features. But you see, there are many, many features which are associated with these MSAs, with these aligned sequences, and also with the target sequence. And then there are these paired representations. Okay. So the purple ones are basically MSA feature. These are extra sequences. These are clustered sequences. The yellow ones basically are the original input sequence based features. Okay. Some of them I'll go into details. I'll at least talk about the feature types, main feature types, right? And then these green ones are basically, features obtained from the template sequences or those sequences which have similar, paired feature from which you get paired features. Okay. Now, we already talked about what you can obtain from the original sequence. You can obtain these AA type features, but you can also obtain some relative positions. Okay, this rel-pos kind of operator actually takes the original, 256 residues and it tries to figure out the relative position of these residues in the original protein sequence. Remember, your original protein sequence could be thousands of amino acids long, but you were able to only take a sequence of 256. So what is the relative position of these each of those amino acids in the original sequence? That is basically what is captured in these kinds of features. Okay. Now these features interact in fancy ways so as to essentially give the final output to the main evoformer stack. Okay, the main input to the evoformer stack. Okay. So, as you see, there are these, dense layers. So they're represented using linear layers. They're mapping. So let me just talk about this target features first, right? So this target features basically is R cross F matrix. What is R cross F? R is the number of residues. So, in this paper, actually, they use R for number of residues as well as N res for number of residues. Okay. And they fix it to 256 basically for pre-training. So that's that. So don't get confused. R is the same as N res. Okay. And SC is the same as clustered and clust. So, and SE is the same as N extra sequences. Okay. So unfortunately, they use two different notations and we can't do much about it. Okay. That's fine. So that's that. So now these linear layers basically are just dense layers. This is the input size and this is the output size. Okay. So they basically do this projection in some ways. And then there is essentially some addition of features happening. There is also, sometimes essentially at the end of it, after all of these interactions that happen, at the end of it, you're going to give the evoformer stack two kind of inputs. One, basically, which is of the size, RRC, which is paired representations. This paired representation matrix is basically going to be here, of that size, RRC. There's going to be residues into C, the number of features. Okay. And then, the other matrix that is going to go is basically SRC, which is number of sequences, number of residues, and the depth of C. Okay. Well, the C could differ in both of them. So it could be RRC1 and, SRC2, but that's basically the two inputs that go to the evoformer stack. Okay. The third thing, so basically we talked about the yellow thing. We also talked about the purple thing. The yellow thing basically is these A type features. The purple thing is these extra MSA features or the MSA features. And as you see here, the extra MSA's number of sequences could be different. The number of clustered sequences are different. You just basically combine them in various fancy ways. You see they're basically getting combined in various fancy ways, and also, followed by the stack, which I'm not going to go into detail. Basically, the stack by itself is a few layers, I think about 8 layers or so. So I'm not going to go into that. But overall, they basically give those two kinds of inputs, a paired representation input, and then an input which basically represents features for every sequence, every residue in every sequence. Okay, that's that. I did not mention about what are the paired features that you obtain. So let's talk about the paired features now, right? So these green features are basically which are represented here. So there are several kinds of paired features. So for example, there could just be a template AA type. So paired features, again, of course, so paired features come from template sequences, and template sequences themselves have uh, you see the SR in the sense that there is a ST kind of number of template sequences, and they have number of residues, 256, and they have their features also. So of course, there's going to be one hot representation for these template sequences, which is basically the template A type. But then there are going to be paired features also. So paired features are towards the end. Let's see. So here are the paired features of template histogram. Okay. So what is this? So histogram is a distance histogram. Okay. Really think of histogram as a distance histogram. So what are those? So basically, you essentially take the beta carbon atoms in every residue. So there are these beta carbon atoms in every residue, and you basically compare the beta carbon of this residue with the beta carbon of that residue. You compute the distance between those in angstroms, okay? And then you map them to one of the 38 pins. So this distance is basically, of course, you can keep the actual distance, but what you do is to convert this feature into a categorical feature. So for every pair of residues, what you have is the discretized distance between their beta carbon atoms, and discretized into 38 pins, and that's basically what you put into this paired matrix. Okay, so for every template sequence, and the for every pair of template sequences, you will basically have essentially residue cross residue paired matrix, which captures these histogram kind of feature. Similarly, you have other features. Again, I'm not going to go into details about other features, but there are many, many other features. Some of them basically are of the shape S cross R. So basically, for every template sequence and every residue, there is a feature. Some of them are R cross R, which is basically for every pair of residues in these template sequences, you essentially have, a feature defined. Okay. So anyway, those features basically go as inputs to our EO stack. Okay. So our Evo Farmer stack basically is going to take these two inputs, the MSC representation and the paired representation. Okay. Again, to stress, the size of this MSC representation is SRC, and the paired representation size RRC. Okay. And the C could be different, basically. Okay. So now the Evo Farmer stack is a 48 block stack without any weight sharing. So essentially, all these 48 layers have their own weights, no sharing of weights. Okay. And essentially, internally, they have this interesting architecture, very interesting architecture. So it's a two-tower architecture in that sense, as you see, right? One tower sort of processes the MSA representation, the other tower processes the pair representation. And of course, the goal finally is going to be to predict protein structure, as a 3D problem, 3D protein structure problem, right? So the AlphaFold model practically, of course, contains two blocks mainly, EvoFarmer and the structure block. And we'll talk about EvoFarmer first and structure later. The overall goal is to be able to convert this problem of finding the 3D structure as a graph inference problem, where you want to find the position of those nodes, and then the position, and then basically, some information about those edges. So for example, what is the length of the edge and so on, right? So you want to basically figure out the nodes and the edges. Nodes basically are primarily represented using residues. Now, of course, you would think about residues, but clearly you would rather represent these nodes as atoms. But remember, these residues basically are mainly represented by the C alpha atom, because the backbone is basically NC alpha C, and then there are side chains. So every residue basically has a NC alpha C backbone and a side chain, and then the C alpha atom can be basically represented as a node in this graph, right? That's that. Now the paired representations RRC essentially are in this matrix, and what you want to basically figure out is this graph. So you want to figure out these IJK, which are basically residues, and you also want to figure out these edges, which are where features are essentially there in these paired representations. Now, the Evo former guy is basically like an embedding guy. Okay, what it tries to do is basically try and learn really good embeddings of these nodes and really good embeddings of these edges. That's what it does. Okay, so in this graph, MSE representation is basically node representation, because it's sort of trying to figure out really good representation for these. So by the way, the first sequence actually is the input sequence in this MSA's first row, and the other rows are aligned rows, right? So what you're going to do is to basically compute a really good representation of the first row and the aligned rows. And of course, at the end of it, you will throw away the representation of the aligned rows, because all you care about is basically a good representation for every residue in the current sequence, so that you can place them as nodes. And then you could basically use the information in these paired representation to figure out the properties of the edges. Okay. But the goal of the Evo guy is to really come up with a good representation for these nodes and edges. Okay. Now, so MSA representation updates the pair representation. So essentially, as you see in these two tower architecture, both the towers are sort of working on their own, but there are some interactions. So in the beginning, the pair representation helps to get a better MSA representation, but later, the MSA representation also helps to get a better pair representation. They talk to each other, right? And they talk to each other in each of those 48 blocks, by the way. Okay, they don't just talk at the end; they talk to each other in each of the 48 blocks. But in some ways, it's still a separate chain or separate tower. There's a MSA tower and the pair representation tower. Okay. When the pair representation, in fact, if you see, there are these two kinds of updates, the orange update and of course, the reddish kind of update. And we'll talk about these updates in more detail. In fact, each of them are like very interesting architectures by themselves. We'll talk about them. Okay. So for an amino acid, basically, if you see these paired representations, right, what do they do? I mean, broadly, what are these layers meant to do? Right? These layers are basically meant to ensure that the final representation that you get enables you to come up with a 3D structure which follows physical constraints. Okay? For example, a triangle inequality should be satisfied, right? So basically, if you predicted that the distance between carbon and nitrogen atom should be this, and carbon and carbon C alpha atom should be that, and N and C alpha atom should be that, of course, they should make a triangle, right? I mean, you cannot basically predict something which is not making a triangle, right? So that's that. The triangle inequality constraints have to be triangle completion constraints have to be satisfied. There are also other kinds of things that need to be satisfied. For example, you cannot have collision of the atoms, right? You cannot basically say, oh, one atom and the other atom are at the same place. You can't have that. So therefore, these layers basically try to ensure those kinds of things. Okay. So let's go into details of each of those layers. And by the way, in the MSA representation, also there are these interesting layers, row-wise gated self-attention. So with pair bias, because it's taking pair representations bias there, and then there's column-wise gated self-attention. So we'll basically talk about those. Of course, they basically follow the same self-attention principles, QKV. It's just that how do you define queries, keys, and values is what differs in these architectures. Okay. So let's get into them one by one. Okay. So first I'm going to talk about this block right there at the top. So the row-wise gated self-attention with pair bias. Okay. So what does this look like? And this is basically how it looks like. It's a complicated block. It looks complicated. It's not really that complicated. Let's try to understand. Okay. So this block basically is going to take both the inputs, the MSA and the pair representation input. Yeah, right? I mean, it's going to take the MSA representation and the pair representation. Okay. So it takes both the inputs. And what does it do? Of course, remember, it's basically SRC, okay? And we'll call it CM. I mean, just to basically say it's the depth of the MSA block, and CZ, basically the depth of the pair block, okay? Now, of course, it's applied row by row because it says row-wise gated self-attention, okay? So whatever you see here is going to be applied for all the rows. So you can take this row and you can basically see that it's basically going to give you a transformed row, a new row, right? And it's going to do this independently for each of the rows here. Okay. So now if you really look at it, this row, what is this row really, right? The dimension of this row is R cross CM, as it says, R cross CM, right? So basically, number of residues cross CM. You can think about every residue is like a token, right? So if you're really used to the transformer kind of thing in NLP, now you basically have a CM dimensional representation of R tokens, and it just do self-attention, right? That's all it does. Basically, that is exactly what is going on. Okay, so you basically take this row, compute query keys and value. These are queries, these are keys, right? Of course, how do you compute queries and keys? I'm not going to go into that. You know how transformer works, so that's basically that. Okay, of course, you also get values. So there are these QKV matrices, and they give you queries, keys, and values. Okay. And of course, typically in self-attention, you do QK transpose. So that is what is going on here. So QK transpose has happened here. And you get this similarity matrix, right? QK transpose. You, of course, do softmax on top of it. Now, the interesting part is that if you really look at it in this world, QK transpose is basically R matrix. It's basically a, RQ, RV matrix. Sure, it's basically RQ and RV matrix. And by the way, the keys are also of the dimensions V's. So keys and values share the same dimension V, RV. Okay. So basically, it's a RQ, RV matrix. And then, of course, you could basically also have a depth H, because when you're multiplying, you can basically say, oh, I want to basically change the depth also. Okay. So you basically have these RQRVH matrix, and you actually had the pair representation here. Okay, so remember, this matrix is more like residues interacting with each other, and you already have a paired representation which you also add. That is how you make use of the pair bias. And remember, the pairs are basically, pair representation is biasing your dot product computation. So it's basically like a pair bias, a pair biased dot product computation. And after you've done that, of course, you apply softmax, like typical self-attention, and you have the attention weights, you multiply by values, you essentially get the output. Now, this is also gated. You know what is gated self-attention? You basically compute how much importance I should be getting from each of my neighbors, right? Of course, self-attention is basically getting information from each of the neighbors. There's gate, which is also computed based on the original representation, decides how much importance should I basically be giving to each of my other neighbors, each of the other residues that are there in the same sequence. And that basically gives me the upgraded sequence. Okay. So what does this row-wise gated self-attention with pair bias do? Well, it's basically going to get you a representation, MSA representation of the same residue, of the same sequence by looking at other residues, and also with the pair bias. Okay, now that's that. Now let's talk about the second block here. Okay, I'm going block by block, and there'll be a slide for every block. Let's think about it like that. Okay. Let's look at the other column-wise gated self-attention. What does this guy do? Now you can imagine if the row-wise basically was working row-wise, column-wise would work column-wise, right? So column-wise guy is basically going to do the same business, but in a column-wise manner. And as you can imagine, the column-wise attention is going to let the elements that belong to the same position interact with each other. Okay. So basically, if this was a residue at a particular position, and of course, it's an aligned residue, right? Not like you've put the residues in a random, put these sequences in a random way, right? They're aligned sequences. Okay. So basically, when you align them, right, you basically can learn from the aligned position from other sequences some sort of a representation for these residues. Okay. So, but otherwise, the entire thing is basically the same gated self-attention which is going on. There is no pair bias here because remember, you're basically doing it for the same position for a sequence that matches across for a residue that matches across sequences, and therefore there's no pair bias here. Okay, but otherwise it's basically the same self-attention, queries, keys, values with the attention weight, softmax, and all of that with gating as well. Okay. That's that. Now, the third block there is the transition block. So the transition block is basically just like your, just like your feed forward kind of a sublayer in a typical transformer. So it's basically just feed forward, right? So it's basically you have the MSA representation, and you basically do the 4x increase of the size of the hidden layer size, right? And then you apply ReLU, another feed forward, another dense layer, and then that gives you your MSA, revised MSA representation, transformed MSA representation. Okay, basically it's just a feed forward layer in that sense. Okay. So once that is done, you see we are done with this tower. So there's an outer product mean tower, which basically gives you MSA. I mean, of course, this gives you your revised MSA representation, but it also gives you input to this outer product mean. So outer product mean basically at the output must give you a pair representation, which can be added with the original pair representation, and then further updates can be done. So what does this outer product mean do? Okay, let's think about it. So outer product mean basically is this network at the right side, right bottom. Okay, it basically is going to take your MSA representation and going to give you the paired representation. Okay, let's see how this is going to do that. Okay. You take these two columns, I and j, and remember this dimensionality is SRCM, right? You take these columns, I and j, you project the CM to C, which basically means that, for both of these columns, what you're left with is, S cross C matrix, right? This is the S cross C matrix, this is another S cross C matrix, okay? You do an outer product, and that finally leads to you outer product. Remember in matrices, the way outer product happens is, you essentially, if you have a vector 1 cross C and another vector C cross 1, you essentially multiply with that and you end up with a C cross C thing, right? The same thing happens here. So you basically align on the S dimension. So there's a S cross C here, another S cross C here. You do the outer product, you get a SCC, okay? So, and then you take a mean across sequences, and that gives you a CC. And then you basically, basically do a dense layer on top of these C cross C features, just to get one single, basically one single vector of size CZ. Okay. For every pair of columns I and J, you basically get a vector CZ, and that's what goes and sits there. Okay. So you basically, of course, do that for all of those I and J's, and that's how you get this paired representation. Okay. So what are you going to do here? You're basically taking the MSE representation and converting it to a paired representation kind of a format. And the way you do that is basically by taking a pair of columns from here and then, doing these transformations as to finally get a RRCZ. Okay. Now, of course, this RRCZ is going to basically get added to the original pair representation and undergo these triangle updates. So we'll talk about these triangle updates one by one, and that will basically finish off our evoformer architecture. Okay. So let's basically talk about those. So how do these various updates happen? So what are these triangular multiplicative updates? Let's talk about that. So remember, the input is pair representation. The output again is going to be pair representation. The size is going to remain the same. Okay. So what happens here is the question. Okay. And let's intuitively understand what is this triangular multiplicative update. Okay. So triangular multiplicative update, basically, there are two of those updates which are going on. Let me just show it to you on the slide. Outgoing edges and incoming edges, two updates based on outgoing edges and incoming edges. Okay, so let's talk about them from the perspective of outgoing edges. Okay, so if I care about a particular node I, right, a node I, then outgoing edges are all represented using the same. In the graph, right, in the graph represented by this matrix, outgoing edges are represented by this row, right? Those are the outgoing edges. Okay. And what the triangular multiplicative update for outgoing edges is going to do is to basically, update the pair representation for this edge ij by considering information from other edges in the triangle where the edge ij where the node I participates. So if j is basically, this is edge ij, which basically means that the original node was I and the destination node was j. So therefore, let's look at all other outgoing edges from I, which is basically IK, right? All of those are IK. So you basically look at those triangles. These are outgoing edges, right? So if IJ was the original edge, you want to update the information there. You basically look at other outgoing edges IK, and then you update IJ based on IK's information and JK's information. You take both of the information and you update the information in the edge IJ, the features for the edge IJ. Okay. For incoming guys, you basically, the way you would do this is as follows. If ij is your incoming edge, right, you basically look for other incoming edges like ki. Remember, I is your central node. So look at incoming edges, ki, and you basically then you also, use the information from ki and kj so as to be able to update the information for ij. Okay, so that's that. So now this one basically shows you processing that you need to do for outgoing edges, and therefore it's like row wise. But for incoming edges, the architecture will be similar but column wise. Okay. Now what is being done here? What is being done is more details in the architecture, but the broad idea is that you're going to take this information and going to update the information, the features for this particular edge by depending on the features of so this is ij, so depending on the features of ik and jk for every k. Okay, so maybe this node is connected to 5 other nodes. So if it's connected to 5, rather, in fact, in general, it's connected to all the other nodes, because there is no notion of an incomplete graph. This graph is fully complete. Every node is connected to every other node. So you're basically going to actually update this information basically using edges, all possible edges, where k goes from 1 to 256 in that sense. Okay. So that's that. Now, for the sake of notation, ik edges are called left edges, jk edges are called as right edges when you consider the node I and you consider the edge ij. Okay. So for edge ij, which is highlighted here, you're basically going to do, linear transformations. So this is a CZ shaped vector. You're going to create a C shaped vector out of it. Okay. So that gives you a C shaped vector. And that's a C shaped vector. Of course, it's a vector for all the residues. So essentially, if you consider the row, you're basically going to get a C-shaped vector for all the residues in this row. So therefore, an R cross C matrix. And you take the C-shaped vector, you compute some number out of it, or rather some C-shaped representation, another C-shaped representation here, and then you multiply the two, you element-wise multiply all of them, you take a sum over R, so that finally you end up with a C-shaped thing, because this is R cross C, so R cross C, you basically take sum over R, so it's to basically get a C-shaped thing. And you convert it again into a CZ kind of a linear transformation, which basically matches with the CZ dimensionality here. You again, also apply a similar gated kind of a thing, which is derived from the original representation. And you multiply this new representation obtained using information from left and right edges to get a revised representation, CZ representation at this position. Okay, that's how you basically get the revised representation. That's a multiplicative update, right? Of course, this is basically showing you how to get the revised representation by making use of this row and the kth row. And of course, you do it for all the k rows, right? All the other rows one by one, so as to get a revised representation here. Okay. Same thing is basically done for a pair of columns, basically called as the multiplicative update using incoming edges. Okay. So that helps us understand this part of the architecture. Now let's basically talk about the triangular self-attention part. There's something called as triangular self-attention also. Let's talk about that now. Okay, how does that work and what does it do? Okay, this triangular self-attention basically is a node-focused thing, and it's also of two types. Let me go back. It's also of two types: around the starting node and around the ending node. Okay. The triangular self-attention is around the starting node and around the ending node. Okay. And the way it basically works is to basically, here basically we're showing the starting node version. Okay. Of course, there's a starting node version and there's an ending node version. Okay. The starting node version, as you see here, it updates the edge information ij with values from all edges that share the same I node. Okay. So it's going to update basically not using jk, right? But using ik, ik1, ik2, and ik3, and so on. Okay. So how much input it's basically going to, how much of information it's going to get from ik, right, is basically going to be derived, of course, based on the similarity, qk similarity, but it's also going to be derived from jk. So it's going to use jk to define the gating information, basically how much information transfer should happen between ik and ij. But but it's going to update the representation for ij based on all other outgoing edges or starting node, basically all other outgoing edges from I. Okay. So remember, this is slightly different from the previous one, because in the previous one, you're basically depending on pairs of edges at a time, so as to define the update to ij, right? But here you're basically going to you're going to do the update altogether for all of those edges which outgo from I, but you're going to define the gating function. The gating function here by looking at the features of the JK edge. Okay. So that's that. So let's basically therefore look at it. So you have the original matrix and you're essentially doing QKV. So and by the way, essentially you take the right edges and essentially use that to create this RQ cross RV kind of a matrix. Okay. And you essentially take the central edge, and you essentially compute the queries. Okay. You take the left edges and you create the keys based on them. Okay. So essentially, you don't do QKV just like that. You essentially do Q based on the central edge. What is the central edge? The edge ij. Okay, you take the row ij, you compute the Q based on that. You take the left edges, which is basically ik edges, and you compute the keys based on that. You do dot product, and you take the right edges basically, and you essentially compute the pair bias in some ways based on that, right? So you add that to the dot product of UK, you essentially get the attention weights from the left edges. Essentially, those are the attention weights that you have. And once you have the final attention weights, you actually have the gating function, which is controlled by the central edges. So you basically take this gating, and you finally get the get the update to the pair representation. Okay, so this update is basically keeping in mind how you can update the values, the features of an edge based on other edges which also start from the same node. But you can also update the information in these edges based on other edges which end in the same node. And then you would basically think about this matrix as columns. Okay. And that's not what I'm showing here, but you can think about that architecture in a similar way. Okay. So that's for the first part of this tutorial on AlphaFold. Okay, so what you've understood so far is basically how Evoformer works. But in the second part, I will basically talk about how essentially the remaining part of the network, essentially, you see the second part, the structure block works, and then how both of them tie together so as to predict the atom distribution or the 3D structure, 3D atomic structure of the entire protein molecule. Okay, that's it for this first part of the video. Hope you liked it, and hope you find it very useful to be able to understand how AlphaFold works. AlphaFold is a complicated architecture, and I've tried to basically explain that architecture in this video and the next. Okay, thank you.